Hello, I'm Dr. Charles Knight, and this is my 10 Lectures on Evolution series. I'm from Cal Poly in San Luis Obispo, California, and I work in the Bailey College of Science and Mathematics and the Department of Biological Sciences. This lecture was originally produced for my Bio 414 evolution class in the winter quarter of 2025, and I posted on YouTube for all to enjoy and learn from. So here we go. Also in this presentation I use our textbook, Evolutionary Analysis, using the 5th edition. Also Wikipedia, Google, as fraught as that might be these days. And also images from the textbooks or books that the authors produced. Two of the authors have new books out, so just due diligence. Most of the images are sourced from these sources. So the paper I'm presenting today is titled Genome Editing Retraces the Evolution of Toxin Resistance in the Monarch Butterfly. It was published in Nature in 2019, and it's one of the few papers from the 20-teens that I presented this quarter. The other one was Jay Stortz's hemoglobin convergence paper from 2018. All of the other papers are from 2024 or 2025. But this one is such an interesting story. I had to keep it, wanted to do a full presentation on YouTube for my students to learn from. It tells a fascinating story that's complicated, but through a lot of very difficult work that was produced by all of these people. I'm going to talk mostly about these 3 investigators, but there's a whole bunch of other people that were involved, and I apologize to them for not doing a full introduction of them. I don't know any of these investigators other than by a few little interactions by Twitter and email, but all of these people have done some incredible work. So I'd like to always start with some scientist vignettes at the beginning. Marianthi Karageorgi was the lead author on this paper, and she was originally born in Greece and then went on to, I know she worked with Noah at UC Berkeley, where Noah Whiteman works now, but currently is at Stanford with Dmitry Petrov, one of my former advisors. And kudos to you, Marianthi, for shepherding this project through to completion. It's a gargantuan, huge amount of work that tells a fascinating story. And Anurag Agarwal, the L is cut off there, sorry Anurag, but Anurag has been a peer of mine since his graduate student days. We were contemporaries. It seemed like every month there was another paper by Anurag in various journals, and we were all wondering how we could keep up with Anurag and his prolific publication, which is kept up while he's still a professor at Cornell and is continuing to produce a fascinating body of research on monarchs and milkweed. He published recently this book, Monarchs and Milkweed, which I just ordered another copy. It's just over my shoulder here. It's a adage that the good books are gone. They're either lent out or they get legs. They are stolen or whatever. I just today received a new copy in the mail. I used this book in a previous class. It's richly illustrated, and many of the images that I'll show today in this presentation come from Anurag's book here, which is one of the most beautiful science books that I know of. Frankly, it's quite beautiful and contains quite a story. Also, Noah Whiteman was the co-PI, I would say probably the PI that got the money to do this work, and maybe was conceived of the idea. An incredible, incredible story. He also published a book recently called Most Delicious Poison, which dwells on the secondary metabolites that plants produce and the ways that insects can tolerate them, and also some personal vignettes on his family's struggles with various poisons like AUD, alcohol use disorder, and whatnot. But I really, really enjoyed this book, which I used for a graduate seminar last year, and it was a great book. So in this presentation, we're mostly going to be talking about the paper, but there's some background information that I want to start with that kind of lays out the story, which is well told in Monarchs and Milkweed. But first, I always like to start with the take-home messages from the paper. I'll read the title again and then go into my 5 take-home points. Genome editing retraces the evolution of toxin resistance in the monarch butterfly. Ouabain is a chemical that disrupts sodium-potassium pumps, which are ATPases. They use ATP to achieve this ion regulation. Monarch butterflies bioaccumulate Ouabain from milkweed plants as caterpillars, and they can tolerate and sequester it so that they become poisonous later in life. That's the classic monarch butterfly story. There's been parallel mutations at 3 amino acid positions in the sodium-potassium pump that lead to Ouabain tolerance across the phylogeny or the group of arthropods. This has happened repeatedly in a convergent way. We could say that these mutations are homoplasious, that is, they're similar but not due to a common ancestor. That's homoplasy. So parallel, convergent, and homoplasious all mean the same thing. These mutations have been produced or have been fixed 3 times, 3 independent times, in 3 different amino acid positions, 21 independent times actually, as we'll see. They took it to the next step by producing CRISPR flies, actually flies, not caterpillars, but flies with these 3 amino acid substitutions that were tolerant to and could sequester Ouabain. So they produced these genetically modified flies that could do the same thing that the monarch caterpillars and butterflies could do. So lastly, this study demonstrates the elimination of a conflicting pleiotropic condition through epistasis. And that's going to be a take-home point from the abstract that I'm going to come back to a couple times, and especially at the very end of this presentation. It's a complicated statement, and I'm going to try to decompress that. So Ouabain is a chemical that some plants make. It's a secondary metabolite. It disrupts these sodium-potassium pumps and its function in animals. So animals have sodium-potassium pumps, but Ouabain disrupts the function, and that has catastrophic effects for most animals. But monarch caterpillars have evolved resistance to Ouabain along with other arthropods. And they actually can bioaccumulate this Ouabain as they feed on milkweed plants. They accumulate it in their tissues and then become poisonous. They retain that Ouabain as they build a cocoon and metamorphose into a butterfly. So the classic story of monarch butterflies is that they have aposematic coloration, warning that I'm poisonous. And then young or naive scrub jays here are learning about Ouabain and feeding on a monarch butterfly, but then quickly throwing up. The throw up is right behind my head here. I should have put my picture in a different spot. Ouabain, the poison, is also used by indigenous hunters to put poison on the tips of poison arrows and darts to immobilize prey, and it works quite effectively at that task. Ouabain can also treat heart conditions in humans. It has been in the past. It's not really used that way. Currently, there's better or different drugs, but in the past it was used. You can still purchase it. You can see the different language on that box, but not currently used as far as I could tell in the US. I'm not an expert on that. But let me read the abstract, as I always do, slowly. The authors put a lot of thought into summarizing their work in one dense paragraph, and so I think we should start with that paragraph, the abstract. Genome editing retraces the evolution of toxin resistance in the monarch butterfly. Identifying the genetic mechanisms of adaptation requires euclidation of links between the evolution of DNA sequence, phenotype, and fitness. Convergent evolution can be used as a guide to identify candidate mutations that underlie adaptive traits, and new genome editing technology is facilitating functional validation of these mutations in whole organisms. We combine these approaches to study a classic case of convergence in insects from 6 orders, including the monarch butterfly, Danis plexippus, that have independently evolved to colonize plants that produce cardiac glycoside toxins. Many of these insects involved parallel amino acid substitutions in the alpha subunit of the sodium-potassium pump, the physiological target of cardiac glycosides. Here we describe mutational paths involving 3 repeatedly changing amino acid sites, 111, 119, and 122, in the ATP alpha that are associated with cardiac glycoside specialization. I'm going to come back to these words in my glossary notes page here soon. This is the second part of the abstract. We then perform CRISPR-Cas9 base editing on the native ATP alpha gene in Drosophila melanogaster flies and retrace the mutational path taken across the monarch lineage. We show in vivo, in vitro, and in silico that the path conferred resistance to target site insensitivity to target cardiac glycosides, culminating in a triple mutant monarch fly that were as insensitive to cardiac glycosides as monarch butterflies were, or are. Monarch flies retain small amounts of cardiac glycosides through metamorphosis, a trait that has been optimized in monarch butterflies to deter predators. The order in which the substitutions evolved was explained by amelioration of antagonistic pleiotropy through epistasis. Our study illuminates how the monarch butterfly evolved resistance to a class of plant toxins, eventually becoming unpalatable and changing the nature of species interactions within ecological communities. Here's some more words that I'll add to my glossary notes page. And monarch flies and antagonistic pleiotropy through epistasis. We're going to come back to this statement over and over again. Here are the glossary notes. Cardiac glycosides are heart toxins like Ouabain, the sodium-potassium pump, which is an ATPase that pumps these ions using ATP to alter membrane potential and ionic concentration, and Ouabain interferes with these sodium-potassium pumps. In vivo, in vitro, or in silico just means in living organisms, in test tubes, or in a computer simulation. Monarch flies are CRISPR-altered flies that have the 3 amino acid substitutions that confer Ouabain tolerance in monarchs. Antagonistic pleiotropy is a gene with conflicting effects on fitness at different life stages, usually young versus old stage. It's a topic that comes up in the evolution of aging quite a bit. And epistasis is an important word that's important to know. It's when a gene or mutation influences another gene or pathway. And we're going to come back to that statement at the end of the abstract quite a few times. This is that statement. This is an information-rich sentence from the abstract. The order in which substitutions evolved was explained by amelioration of antagonistic pleiotropy through epistasis. Let me define a few of those words here. Amelioration just means lessening of or resolution to. Ameliorating makes it easier, fixes it, lessens it, resolves it. Second, antagonistic just means conflicting or opposing. Pleiotropy is when one gene or mutation has multiple phenotypic effects. Epistasis is when genes or mutations interact. Amelioration of antagonistic pleiotropy through epistasis means when epistasis resolves a condition of conflicting pleiotropy. And antagonistic pleiotropy it usually refers to a conflicting fitness effect at different life stages, like young versus adult, young versus old. And I have a few more illustrations of that here next. So in our textbook, there's a whole section on trade-offs in aging and antagonistic pleiotropy. This is a nice image that I found on the internet from this website, frozenevolution.com. I don't know anything else about this website other than this useful image where there's a mutated allele that has a positive effect on a juvenile state but a negative effect on the older state. that would be an example of antagonistic pleiotropy. Another pleiotropic condition is sickle cell anemia, where the one mutation in hemoglobin causes the hemoglobin or the red blood cell to sickle in shape, and that leads to sickle cell disease, which is devastating for people that have it. But in the heterozygote state, as you know, that confers some resistance to malaria, and we can see this map of the sickle cell allele in Africa and also the frequency of malaria, and there's a pretty close concordance from where the sickle cell allele occurs and where malaria is high. People that are heterozygotes have some resistance to malaria, and this would be a pleiotropic state of the sickle cell allele. In this case, not too antagonistic, because it confers resistance across the whole life if it's in the heterozygote state, but it's antagonistic between different genotypes. This is the first figure from the paper. Unlike most of the papers that I've presented, I'm actually going to talk about every element of every figure in this paper. It's a Nature paper, so there's only 2 figures really, but each of those figures is very, very information dense. So I'm going to parse out each element of each figure and try to explain it in the most detail that I can, so that you can understand it as best as possible. We'll start with the image of the sodium-potassium pump, this space-filling model showing some alpha helices and whatnot. It's a membrane-spanning protein. You can see Ouabain in the gold in the center in the binding pocket. This is the ATP alpha subunit, which is membrane spanning. The whole protein is about 1,000 amino acids long. We're going to be talking about mutations in this binding pocket next. So they exploded out this binding pocket and show where the Ouabain is nestled in there, and the 3 amino acid mutations that led to Ouabain tolerance. And I'm just going to slow down and write out each one of those. So the first mutation is a Q to V mutation at position 111. And that's a Q to a valine. Valine is the derived amino acid state. The red letters are the derived monarch amino acids at that position. The second mutation is at position 119, which is an alanine to a serine, and the third mutation is at position 122, which is a N to an H. I forget what N amino acid is, but histidine is the derived state. So these 3 amino acid substitutions at 3 different locations in the protein are derived amino acid conditions that lead to Ouabain tolerance. If we look across the phylogeny of insects, we see multiple instances of the evolution of feeding and sequestering Ouabain, and we're going to start by looking at this state of feeding or sequestering. I just exploded it up so you can see a filled green square means that that insect can feed on plants that have Ouabain. Purple filled square means that those insects can sequester the Ouabain so that they become poisonous later in life. Looking across the phylogeny, we can see that Ouabain feeding and sequestering has evolved 21 times independently. Monarchs are just one test case of Ouabain tolerance and sequestering of insects. The thickness of the line that is pointing to the taxa indicates how many of the mutations that insect has. So the thickest line has all 3 mutations, 111, 119, and 122. Medium thicknesses are 2 of the 3 mutations, and light thickness is 1 or at least 111 or 19. And the thinnest line is no change. ASR is ancestral state reconstruction, and so through ancestral state reconstruction and parsimony, we can predict or infer that Ouabain feeding and sequestering occurred 21 times independently in this group of insects. So first I'm going to explode out or focus in on the monarch group. Here we can see the monarch butterfly, Danus plexippus, has the 3 amino acid mutations. Remember, the whole protein is about 1,000 amino acids long. These 3 specific spots are in the Ouabain binding pocket, and the derived state is VSH. If we compare that to heliconius butterflies, they have the ancestral state, which is QAN. And I'm just giving you this QAN again. The ancestral state is QAN. The derived, fully tolerant state of monarch butterflies is VSH in these 3 amino acid positions. This is the second figure, and it's got quite a bit of information in it, and I'm going to try to, again, explain each one of these figures individually. The first figure shows a phylogeny of the monarch group. Notice these are monarch butterflies shown as icons here on the right, with various colors indicating which of the 3 amino acid substitutions they have. The first mutation was a Q to an L at position 111. The second mutation was a A to an S at position 119. The third mutation is at position 111 again, a second mutation, an L to a V, and then the third mutation is at position 122, an N to an H. So it goes in this fashion. We can actually predict that these 3 amino acids these 3 amino acid substitutions had to happen before the genotype VSH evolved, which was fully tolerant to Ouabain. VSH is the fully tolerant genotype, and it's shown in red throughout this presentation. The black QAN is the ancestral state. This figure is quite a lot of information, and I'll start just with the monarch butterfly on the far right hand side. We see the ancestral QAN is mutated to LAN in the monarch, and then to LSN, and then to VSN, and then to VSH, which is the fully tolerant state. Notice the legend here on my right next to me. It looks like the Italian flag, and it just has those 3 mutations in the ATP alpha. If we expand out to the whole monarch group, we see that LSN is a fairly common genotype within Lepidoptera, and we'll see that LSN has an intermediate tolerance to Ouabain. Looking out across, the question is just how do these genotypes compare for Ouabain tolerance, and we'll see that in the physiological assays that they do. Looking across the rest of the phylogeny, we see that VSH has evolved at least 3 independent times, and so it's not just monarch butterflies that have VSH, but other arthropods have different genotypes, some of which confer tolerance and sequestering of Ouabain. Next, we'll jump right into the CRISPR mutant flies. These are flies now. Notice the icons are flies, not monarch butterflies. They're actually flies that have these individual amino acid substitutions. So they did base pair editing to produce these amino acid sequences that were the same as those intermediate steps as the monarch butterflies had. So we're basically retracing this evolutionary trajectory, but in genetically modified flies. They also produce some alternate paths to Ouabain tolerance, and I'm not going to talk a lot about that until it comes out in the physiological assays. Just realize that this gray box indicates some alternative paths. Now we'll jump right into the first of the physiological assays, which is a dose-response curve of Ouabain tolerance for larva to adult survival. Again, the x-axis is Ouabain concentration in the food. Notice their image there on the left is a Drosophila larvae feeding on a mixture that has various concentrations of Ouabain. They've got the chemical signature there or the chemical model in the feed, or in the whatever the gel, the food that they're feeding to the Drosophila larvae. The whole axis, the x-axis, is the dose response curve. The larva to adult survival in percentage is on the y-axis. And QAN star is the wild type genotype, and the CRISPR flies are all of the other genotypes up here. And so those were mutations, base pair editing in the flies to produce these different amino acid genotypes. QAN here is a re-engineered wild type CRISPR control. So we expect that QAN no star and QAN star should perform about the same. Also note that there's a discontinuity on the x-axis. This gray box are Ouabain concentrations that they didn't test. They jumped from 15 out to 30, whereas they had been going up in 1.5 millimolar concentrations from 0 to 15, and then they jumped out to 30. Usually dose response curves are on a logarithmic scale. This is kind of a hybrid linear logarithmic scale. And I'm just highlighting again that these genotypes in the flies are meant to correspond to the genotypes in the monarch butterflies, and so they're trying to retrace the tape of time, recreate it in the genetically modified flies, which is pretty amazing that they were able to do that. Again, notice that gap, the discontinuity. You'll see some of the lines will be offset because it goes across that gray box. So what I want to do first is just predict what the dose response curve would be in the wild type. I expect that with increasing concentration of Ouabain, the survival or larva to adult survival will decrease, and at the highest concentrations it'll be 0. So that's what I've drawn here. 0 larva to adult survival on the highest concentrations, but declining survival with increasing Ouabain concentration. Secondly, in the VSH, Ouabain tolerant genotype from monarch butterflies, we'd expect the monarch flies to also be tolerant. No change in survival, none at all. It can handle any concentration of Ouabain and actually feed on it. Intermediate genotypes will have intermediate phenotypes, that is, intermediate tolerances going down. Notice the discontinuity happens across that gray box because it jumps out to 30, and the last intermediate genotype will be the lowest. That'll be the first mutation. Again, a discontinuity there. Now we'll look at the actual data. Well, actually, first we'll just rank the performance at 30 millimolar, 1, 2, 3, 4, 5, going down the ancestral path back to the ancestral state, being the least tolerant of Ouabain, especially at 30 millimolar. Now we could look at the actual data, and it lines up almost perfectly. These are the real data from the figure I forget what it is, 2C. We also rank the performance there. It has the exact same rank as what we predicted. Notice that there's some other genotypes, these alternative paths, which are kind of confusing because they're in different shades of dark blue, which is difficult to differentiate on the figure. But interestingly, the QAH genotype has high tolerance. That is, it's not that different than VSH. The resolution of that is not well described in the paper. This indicates that there's other ways to get to Ouabain tolerance. It doesn't have to be those exact same 3 mutations. But then QSN is also an intermediate genotype. So it has one. VSH is the genotype of the monarch flies or monarch butterflies. VSH in the flies produced the highest fitness, but VSN produced a little bit of fitness, that is larva to adult survival, and QAH though was 100% tolerant, which is has only one of the same mutations as VSH, which again, is a little bit confusing to me. There's still that discontinuity across that gap. Now we're going to go to adult survival, and these are actual dose response curves for adult survival, and we'll rank them again. 1, VSH is the highest. All of the other genotypes at 30 millimolar were not tolerant. That is, the adults died. They couldn't hang at 30 millimolars. If we look at 9 millimolar, we can see the same rank, 1, 2, 3, 4, 5, going down the same as the rank, at least, was the same as for the larva to adult survival. Again, the alternative states have conflicting but interesting. QAH is still quite tolerant up near VSH, and QSN is kind of back in the pack with the other intermediate genotypes. All right, now we go to another great extension, actually feeding the larva on milkweed extract, not isolated Ouabain, but actually milkweed extract. So you see now the larva is on a green sludge in the feed, and that's milkweed extract. We see the VSH genotype has a higher number than the QAN. The y-axis is change in egg to adult survival on milkweed diet, and that's change from the normal feed for these flies. So the QAN wild type, that's the real wild type flies, had a decline of 25% survival from egg to adult on this milkweed extract. It could tolerate it a little bit. It didn't just up and die, but the VSH had a 18% better survival on the milkweed extract diet. So not only did it survive, but it did better than the control. So it can not only sequester and metabolize this Ouabain, it actually gives it a fitness advantage. The intermediate genotypes had an intermediate level or intermediate change in egg to adult survival. Lastly, from their figures, they show sequestering of this Ouabain. This is a complicated figure. Let me explain it slowly here. There's 4 different genotypes shown here. The wild type fly on the left, all the way up to VSH. The fly Ouabain concentration, the amount of Ouabain in the adult fly after it was exposed to various concentrations of Ouabain in its diet, and it was 6 days. It was on this diet for 6 days, various concentrations. You see on the right-hand side, the size of the dot. Each dot is a fly that survives. The size of the dot is the amount of Ouabain in its diet. We'll look at the VSH first. Bunch of flies survived. Bunch of them survived on high concentrations of Ouabain, and those flies tended to accumulate more Ouabain in the fly. That is, when you ground up the fly and analyze the amount of Ouabain in the adult fly after 6 days of feeding, the ones with the highest amount of Ouabain were fed with the highest amount of Ouabain. Compare that to the wild type flies. Those are the Ouabain. Some VSH genotype flies sequestered a lot of Ouabain. Some of them didn't sequester that much, but those ones may have been reared on less Ouabain. If we look at the wild type fly performance on milkweed diet, we see that the wild type QAN star did not sequester much Ouabain. The ones that were fed on the highest amounts of Ouabain probably didn't survive. We don't see any large circles. The only ones we see are small circles. So a few of them were able to survive, but they didn't sequester much Ouabain. Intermediate genotypes had intermediate performance. So intermediate genotypes were intermediate. So let me try to explain this hard sentence from the abstract again. Remember, the order in which the substitutions evolved was explained by amelioration of antagonistic pleiotropy through epistasis. So these mutations showed an antagonistic pleiotropic condition. And let me explain how that is. These 3, blue, yellow, and orange, are intermediate genotypes. they show antagonistic pleiotropy because in the larval state they were protected, but in the adult state they were not protected as well as the VSH. So I consider this to be antagonistic pleiotropy, where there's a benefit on the larval stage for Ouabain tolerance, but not as much in the adult survival stage. But this third mutation, that position 122 End-to-H mutation, ameliorated the antagonistic pleiotropy because we see that larvae and adults were protected from Ouabain. So that was the amelioration of antagonistic pleiotropy. And I had to explain every figure to get to this point where we could try to see a graphical presentation of this amelioration of antagonistic pleiotropy. Basically, the horizontal red line on both the C and D charts indicates that there's no longer any cost to juvenile survival or adult survival. Larvae and adults were protected. All right, I'll go back to my take-home points. Genome editing retraces the evolution of toxin resistance in the monarch butterfly. First, Ouabain is a chemical that disrupts the sodium-potassium pump, which is an ATPase. Monarch butterflies bioaccumulate Ouabain from milkweed plants as caterpillars, and then they can sequester and retain that later in life. Parallel, convergent, homoplasious mutations at 3 amino acid positions in the sodium-potassium pump led to Ouabain tolerance. GMO, CRISPR, Drosophila with these 3 amino acid substitutions are tolerant to and sequester Ouabain. And lastly, this study demonstrates the elimination of a conflicting pleiotropic condition through epistasis. Thank you very much.